Having left chapter 5, which we just covered on the topic of energy, what energy is, different forms that it occurs in, why it's important to cells, uh, enzymes, ATP, all those were subjects of chapter 5. This chapter is an extension of chapter 5. Chapter 7, titled How Cells Harvest Energy from Food. This covers the specifics of, like the title says, how we take energy from the food molecules that we ingest in our bodies and use that energy to make that molecule called adenosine triphosphate, or ATP for short. ATP is a molecule that all cells on the entire planet utilize as a common currency of energy. That is a molecule that cells use for anything that requires an input of energy, whether it be the movement of the cell, like crawling or swimming with flagella, or inside the cell, building molecules through dehydration synthesis. That takes energy. Or moving a vesicle in a eukaryotic cell as part of the endomembrane system. That requires energy. Virtually everything that a cell does requires energy, and ATP is the molecule that provides that energy for cell activity. So again, this chapter is how we make this ATP using energy from our food. Now I've got an image in the top right of this slide of a newborn baby being held, a little traumatized after coming out of its mother's uterus for, maybe about 9 months. I imagine I was kind of traumatized as well. But ever since this baby was a fertilized egg, a zygote, even before then, as a sperm and egg, she has produced her ATP in her cells using oxygen and sugars from her mother, that she got through her umbilical cord. Now she's living independently of her mother, and she's going to continue to make ATP in the cells of her body to fuel all the cellular activities that are going to help maintain her, help her grow, help her develop. In addition to that, it's been recently found that babies are born with a specific kind of fat called brown fat that uses energy from food molecules not to make ATP, but to release the energy from those food molecules as heat. So this can help a newborn baby stay warm in its new environment outside of its mother. Well, look at the figure on the bottom part of this slide. It's showing you sort of the sequence of energy that happens in ecosystems on the planet. And as the top of the figure is showing, virtually all the energy that life uses on Earth comes from the sun in the form of light. And as that light energy is coming down to our planet, organisms like plants and algae are capturing that light energy and using it during photosynthesis to make food molecules, organic molecules like sugar. And a byproduct of that chemistry of photosynthesis releases oxygen. Now, that's not why plants are doing photosynthesis to release oxygen. They're doing photosynthesis to make food molecules for themselves, organic molecules, again, like sugar. Now, other life forms like us, animals, we need energy too, of course. All life forms need energy, so we eat those food molecules that are made by plants. And those food molecules are processed mostly in our mitochondria. And energy is extracted from our mitochondria, or our mitochondria extract energy from the food molecules to make ATP, that common currency, energy currency of cells. And that process is called cellular respiration. And so that's what you're learning about in this chapter, the details of cellular respiration. as a result of breaking down food molecules to get the energy out, the sugars are broken down into carbon dioxide and water. So those are the products of cellular respiration. The carbon dioxide is what we exhale, and water we can use in our bodies. And there's the cycle. Okay, so carbon dioxide and water is then used by plants to make more sugar using energy from the sun. And then those sugars are used by cells like our own to make ATP, and the cycle continues. Remember the second law of thermodynamics. So as plants and animals and all life forms are utilizing energy from food molecules, some of that energy is lost as heat. And so that we have to keep replenishing energy from outside sources, from the sun, to keep our life going. So let's jump into this chapter. We're going to start off with some kind of general background, a little bit of chemistry, and then we're going to get into the nitty-gritty details of exactly how we extract energy from our food molecules to make ATP. So here we're pointing out, remember from chapter 5 that metabolism was discussed. Metabolism is all the chemical reactions that happen in a cell or an organism. And metabolism is made up of anabolic pathways where bigger, more complex molecules are built, and catabolic pathways where big complex molecules are broken down into smaller ones. Well, during cellular respiration, catabolic pathways are what occur, what happen. Cells take complex organic molecules that are full of energy, and cells break them down into smaller components, specifically carbon dioxide and water. And in the process of a catabolic reaction, energy is released. So here, as energy is released in cells through the breakdown of complex food molecules, that energy is harnessed to make ATP. So that's a general sort of overview of what this slide is all about, but let's start at the top with the first bullet. So cells are non-stop, ceaselessly, never-ending, breaking down or degrading these complex organic molecules that are rich in potential energy, usable energy. And when they're broken down, there are simpler molecules that are produced. These are waste molecules that cells really don't need, and they have less energy in them. And those waste molecules would be carbon dioxide and water. Now I've got a question there in purple. Can all of the released energy that's coming out of these complex molecules be used to do work, like to make ATP? Can 100% of it be used to make ATP? And the answer is no, because of the second law of thermodynamics. Some energy, quite a bit of energy, is always lost as heat, disordered energy. So cellular respiration is not 100% efficient. It's in fact about 34% efficient. Only about 34% of the energy that's in your food molecules actually makes ATP. The other 66 or so percent of the energy in your food leaves your body as heat and helps keep your body warm. So, second bullet, most eukaryotic and many prokaryotic organisms carry out what's called aerobic respiration, the equivalent of cellular respiration, where organic fuel, like sugar, food, and oxygen are the reactants. And the resulting products of this chemical reaction are carbon dioxide and water, and energy is released. So there's a picture of the chemical reaction down there near the bottom of the slide. Organic compounds plus oxygen, they react, carbon dioxide, water, energy is released. Now this is really similar to the combustion of gasoline in your car's engine or in a car's engine. So oxygen is required, and gasoline is mixed with oxygen. It's combusted, a spark plug, and energy is released. Energy that can drive your car. But of course, a lot of the energy that's released from combusting gasoline in your engine is lost as heat. It doesn't drive your car. What comes out of your exhaust pipe? Well, carbon dioxide, which of course is contributing to climate change, and water, which you can sometimes see like dripping out of an exhaust pipe. So essentially the same kind of combustion happens in our cells, except the reactant is glucose down on the bottom, and it reacts with oxygen, and carbon dioxide and water are released, and we use as much of the energy as we can to make ATP. But again, because of the second law of thermodynamics, we lose a lot of the energy as heat. So we have to keep replenishing the energy by eating more food. Now, another thing we need to cover here before we jump into the details of how cellular respiration works is this topic of oxidation and reduction, which is a chemistry topic. And every chemical reaction involves the transfer of one or more electrons from one reactant to another reactant. So electrons are moving around. Chemical reactions involves electrons. We kind of covered that in chapter 2, learning about covalent bonds and ionic bonds and how electrons are moved around. Now, any substance that loses electrons is oxidized. That's the word that chemists use. And the process is called oxidation. Any substance that gains electrons during a chemical reaction is reduced, and the process is called reduction. We use that word, or chemists use that word, reduced, because if you gain electrons, then your positive charge is reduced. Electrons counteract that. An example is given of oxidation and reduction here in the middle of the slide with sodium and chlorine to make sodium chloride. So the sodium becomes oxidized when it loses the one electron that's on the outside of its shell. And chlorine needs an electron in its outside shell. You might remember this from chapter 2. And so it gains that electron that's coming from sodium, so chlorine gets reduced. And as a result, we end up with a positive sodium, because it lost an electron, and a negative chloride, because it gained an electron, all because of oxidation and reduction. Now, just below that, more generally, you could think of any kind of oxidation reduction process in a chemical reaction like, okay, we have substance X that has one or more electrons that are going to be removed, okay? And then we have substance Y that's going to gain those electrons, so it becomes reduced. So you see on the other side of the equation and the products that X no longer has that electron, but instead Y is holding on to it. So that's kind of a general way to look at the process of oxidation and reduction. And we might say that X with electrons is a reducing agent and substance Y is an oxidizing agent, but we don't need to quite get into that here. So anyway, we're going to see, because this is what happens during cellular respiration, is X is basically sort of our glucose, our sugar, and Y is our oxygen. So the sugar is going to lose electrons along the way. Many electrons are going to be taken out, and eventually oxygen is going to grab onto those electrons, and you'll see the details of this as we go on. But all of this is applied to and relevant to the steps of cellular respiration, as we're going to jump into next. So in fact, yes, here we go. This slide is specifically showing you what's being oxidized and what's being reduced during cellular respiration. And the title of the slide, Oxidation of Organic Fuel Molecules During Cellular Respiration. So here's a summary equation on the top, which you should know. You should be able to write it out. You should know it, recognize it. And so we begin with a molecule of glucose, sugar, and the hydrogens and their electrons are going to be removed from that glucose. So the glucose becomes oxidized. And the oxygen that you breathe in and goes to the cells of your body is going to gain those hydrogens and electrons, and it's going to be reduced. And when it becomes reduced, it'll become water. And when the glucose is oxidized and the electrons and hydrogens are removed, then we're left with carbon dioxide. Okay? And energy is released as these electrons are moved around, and that energy is what's used to make ATP. So the second bullet down here is reminding you that of all the elements in the periodic table, oxygen has the second highest electronegativity of all of the elements. It is extremely electronegative. So oxygen is a fantastic substance to be grabbing onto electrons from our food molecules. It naturally has this tendency to want to grab them and take them out. So that's a good thing. That means we can breathe in oxygen. It can go to the cells of our body, and it can help remove electrons from our food so we can get the energy out of our food. That's basically what oxygen is doing for us. Okay, so another thing to remind you of near the bottom of this slide is that every chemical reaction has an activation energy that it has to overcome for the reactants to turn into products. We learned that in chapter 5. And so enzymes are going to be available at every step of the way during cellular respiration to help lower the activation energy and increase the rate of all the chemical reactions. So that's another thing we're going to point out to you, is how along the way during cellular respiration there are enzymes to help lower the activation energy barrier. Okay, moving on. So I told you at the beginning that there were a few chemistry topics that we needed to cover before we could jump into the steps of cellular respiration. And we just talked about oxidation and reduction. So that's a really important chemistry topic to have in the back of your mind as we move on. But here's something else that you kind of need to be aware of before we jump in. And that is that there's a molecule, a cofactor, a coenzyme. Again, that's a word and a topic from chapter 5 that is abbreviated NAD plus, because it's got a positive charge. And this molecule helps to shuttle or move electrons from one place to another. It's like a little taxi cab that, hey, electrons, I'll grab onto you and I'll shuttle you over to this other place. And that's the job of this molecule. In fact, that's the job of many cofactors or coenzymes in cells, is to help move electrons around during chemical reactions. So during cellular respiration, of course, unlike burning gasoline or something where all the energy is released as heat and light, cellular respiration is a very controlled process. There's a whole series of chemical reactions, one after the other, that your cells are carefully controlling. And so the energy is not released in a big explosive step, which would kill your cells and damage cells, but the energy is released in little chunks, little steps. And NAD helps to do that. It helps to move electrons around and release the energy in small steps. So this NAD, by the way, is an abbreviation for a longer name that I won't ask you to write out or probably recognize. And that longer name is nicotinamide dinucleotide. Pooh, yeah, that's a mouthful. And when it gains electrons and hydrogen, when it is reduced, then it becomes NADH, which you can see down on the bottom here. And that NADH then is our shuttle, our taxi cab, that's going to take the electrons someplace where they are needed or wanted. And enzymes that help to remove hydrogen atoms and electrons from molecules are called dehydrogenases. Makes sense. D means without or to take away hydrogen. And of course, ACE is the ending for an enzyme. So you're going to encounter that name, dehydrogenases, if you move on into other biology courses after this. So it's a good thing to know. Dehydrogenases are enzymes that remove hydrogens and their electrons from molecules. So there are dehydrogenases that remove electrons and hydrogens from our glucose, from our organic fuel, and then those enzymes help to transfer those hydrogens and electrons to NAD, our little shuttle, turning it into NADH. Now we're going to see during the process of cellular respiration, as I show you the steps on various slides, that we begin with a molecule of glucose. That's our fuel. The electrons and hydrogens are taken out of that and given to NAD. So now we have NADH, our little shuttle. And then those electrons and hydrogens are going to be moved near the end of cellular respiration to what's called the electron transport chain, abbreviated ETC on the bottom here. And at the very end of the electron transport chain, after the energy from the electrons has been released, oxygen is going to be there to take the electrons out of the electron transport chain and make room for more. So you'll see this. This is sort of your first introduction to this process, and it's being shown to you here because of this topic of electrons being moved around by NADH as kind of a shuttle. Okay, moving on. So I've just reviewed with you some of the chemistry, the background that you need to be exposed to and have a grasp of before we do cellular respiration. Again, we talked about oxidation and reduction, and we just talked about this electron carrier molecule, this little shuttle called NAD that turns into NADH. Now I am showing you sort of a preview of cellular respiration by showing you the major stages, the major chemical stages that happen during cellular respiration that help us make all this ATP. And if you look at the picture, look at the figure down on the bottom right. Again, this picture is loaded with details that are all important. First of all, you're being shown that, hey, this happens in mitochondria of animal cells. This happens in plant cells and in their mitochondria. So all eukaryotic cells are doing this. It's an important thing to realize, including our own cells in our bodies, because we are animals. And the first chemical process to occur during cellular respiration is called glycolysis. So you can see that in the figure. It says stage 1, glycolysis. And glycolysis, where does it happen? Where does it occur in your cells? Well, look at the figure there. The cytosol, okay, that liquidy part of your cells that surrounds your organelles. That's where glycolysis occurs. And it begins, what does glycolysis begin with? What's the starting molecule? Okay, that again is in the figure, glucose. And during glycolysis, glucose is oxidized. That means we're taking out electrons and hydrogen. And those electrons and hydrogens are given to NADH, okay, which you can see in the diagram on the bottom. The NADHs are shuttling, and another molecule called FADH2 that you'll encounter a little bit later here. They are both shuttling electrons down to that electron transport chain that's down here. So at the end of glycolysis, there's about 10 chemical reaction steps then what we're left with is two molecules called pyruvate. And those molecules then are going to enter into the mitochondria, and we're going to end up with stage 2 of cellular respiration. This is really where aerobic respiration is happening, because this is happening in the mitochondria and it requires oxygen. So that's why it's called aerobic respiration, because it requires oxygen. And in stage 2, we have pyruvate oxidation and the citric acid cycle. So, as the name implies, pyruvate oxidation. These pyruvate molecules are oxidized, which means that electrons and hydrogens are taken out, they're given to NADs, and they turn into NADHs, which are shuttled to the electron transport chain. As a result of taking out those electrons and hydrogens, carbon dioxide is produced, part of our waste. Now, in the citric acid cycle, the remaining electrons and hydrogens are taken out of our fuel molecule, completely stripping them out and giving those electrons and hydrogens to our little shuttle molecules, and a little bit of ATP is made. Only 2, and by the way, 2 ATPs are gained by glycolysis as well. So a little bit of ATP is made along the way in this chemical pathway. The last phase, stage 3 of cellular respiration, is called oxidative phosphorylation. It's quite a mouthful. And basically it uses the energy released when NADHs and FADH2s pass their electrons on to oxygen through the electron transport chain. And so the energy is released from all these saved up electrons. That's the crux of this. That's the important thing. All the electrons are released from NADH and this other thing called FADH2, and the electron energy is used to make ATP. So you can see what I wrote here, about 90% of the energy that we end up using to make ATP is released and used down at this oxidative phosphorylation step. So that's where the vast majority of ATP is made, is in the very last steps of this process. Okay, well again, this is a general background showing you the major stages of cellular respiration for you to start to catalog in your brain, and to show you where they occur. Glycolysis in the cytosol. Everything else is inside our mitochondria. And that through the chemistry of all this, electrons are being taken out of our fuel molecules and shuttled to this oxidative phosphorylation process to make the vast majority of ATP. All right, so here we go. We're now going to jump into the nitty-gritty of how all this chemistry works to make your ATP that powers your cells. And like I said on the previous slide, the first stage is called glycolysis. And glycolysis is made up of about 10 chemical reaction steps. And the first 1, 2, 3, 4 steps, 5 steps are called the energy investment phase. Okay, the energy investment phase of glycolysis. And the reason why these first series of chemical reactions are called the energy investment phase is because 2 molecules of ATP in 2 different places here are used as an energy source to kind of get these chemical reactions going, to get them started. It's kind of like lighting a match to get a fire burning. So energy has to be invested to start the cascade of chemical reactions that are going to release energy and make ATP. So that's why it's called energy investment. That's what we're beginning with. Yeah. First, the very first chemical reaction step begins with glucose, C6H12O6. And a reminder, we are after all the electrons that these hydrogens, these 12 hydrogens are hanging on to. That's what we're after. That's what's going to be removed in gradual steps. And those electrons are going to be saved up and carried by NADH and FADH2 to make a whole bunch of ATP at the end. So we begin with a molecule of glucose in the cytosol. 2 molecules of ATP are invested to help destabilize our glucose and split it into 2 smaller sugars called glyceraldehyde. And that's what you see at the very end here before step 6, is this little molecule is a sugar, a 3-carbon sugar called glyceraldehyde. And there's 2 of them. And each one of them has a phosphate group attached to it, phosphates that came from these ATPs. And because the phosphates are attached to the number 3 carbon in the glyceraldehyde, chemists call it glyceraldehyde 3-phosphate. It's the origin of the name. And many of us, including myself, refer to it as G3P for short, glyceraldehyde 3-phosphate. So that's the end of the energy investment phase. This figure here is showing you these steps. Now there's much more in this figure than what you need to know. What you need to know is highlighted in my text on the slide, and I just reviewed it with you. But I would like to point out to you that along each step of the way, as I mentioned, there's an enzyme to help lower the activation energy and increase the rate of all these chemical reactions. And remember, enzymes are specific to certain substrates. So hexokinase, you don't need to know these enzymes, you guys, but I'm just pointing this important concept out. Glucose fits into the active site of hexokinase, and so does ATP. And we get a chemical reaction, and out pops glucose 6-phosphate. Then that molecule enters into the next enzyme, which you don't need to know the name of, phosphoglucoisomerase. These enzymes are named after what they're doing. And we end up with a molecule that pops out of the active site called fructose 6-phosphate. Then that enters into the active site of the next enzyme with the next ATP, and so it goes. My main point being there's an enzyme for every step of this metabolic pathway to help speed it up. So here we're looking at the second half of glycolysis. Remember, there's about 10 chemical reaction steps, and the first half we just covered called the energy investment phase. The second half is called the energy payoff phase, because now we're going to start extracting energy from our fuel molecules and starting to use that energy to make a little bit of ATP. So we're starting to get a payoff. So let's check out the details. The details of what you need to know are summarized as usual on this slide. So the beginning of the energy payoff phase involves this glyceraldehyde 3-phosphate, G3P, that we just finished discussing on the last slide. And there's a couple important things that happen right away with that G3P. It fits into an active site of an enzyme. You don't need to know the name of it, but so does an NAD. And 2 electrons and a hydrogen are removed from that G3P and given to the NAD, and it turns into NADH, pops out of the enzyme, and is now shuttling and carrying those electrons and hydrogen to where they need to go. There's a phosphate group that's pulled out of the cytosol, and it attaches to this G3P molecule. And now we have 2 phosphates attached to that molecule. Okay, and we go on from here. So again, to quickly summarize this first part of the energy payoff phase, G3P is oxidized and NAD is reduced. And as a result, our G3P has 2 fewer electrons and 1 fewer hydrogen than it had before. And a phosphate group is pulled out of the cytosol and attached to the remaining molecule, and that's what we have. Now, in 2 separate chemical reactions, different steps along the energy payoff phase, those phosphates that are attached to our fuel molecule are transferred to ADP to make ATP. So we're getting some payoff. We're getting some energy payoff here. Once again, this substrate with phosphates goes into the active site of an enzyme, along with an ADP fitting into the active site, and the phosphate transfers from the substrate to the ADP, and out pops ATP. This happens in two different places. And this process by which the ATP is being made like this, using an enzyme to help transfer phosphate groups, is called substrate level phosphorylation. The next slide shows you a picture of how this happens. Now, at the very end of glycolysis, at the end of these 10 chemical reaction steps, we no longer have glucose, of course. We haven't had glucose for a long time because the sugar molecule keeps getting modified. But we have is a molecule of pyruvate. Now, notice that there's a number 2 in front of all these things. 2, 2, 2, 2. Why is that? That's because there are 2 molecules of glyceraldehyde 3-phosphate that are coming through these chemical reactions. We started with 1 glucose, and it got split into 2 glyceraldehyde 3-phosphates. So each of those G3Ps are moving through this conveyor belt of chemical reactions. So as a result, there are 2 NADHs that are made. At this chemical step, there are 2 ATPs that are made because there's 2 molecules moving through here. So double. There are 2 pyruvates at the end. Again, there's a specific enzyme along each step of the way. Just pointing that out here for you to understand. You don't need to know the names of all these enzymes. Now, if we look at the very bottom of my slide, I'm summarizing. What are our overall inputs in glycolysis? Well, an input was a molecule of glucose at the very beginning of the energy investment phase. An input, 2 molecules of ATP, and an input, 2 molecules of NAD. Right. Well, what are the outputs? What are we getting out of all this? Well, we're getting 2 molecules of pyruvate. That's an output. We're getting 4 ATPs being made, 2 here and 2 here, but we have a net gain of 2. Why? Because we invested 2 in the energy investment phase. So if you invest 2 and you get out 4, that means you gained 2. You have a net gain of 2. And finally, what's the last output? 2 NADHs. Those are our electron carriers. So this is a summary, again, of what you need to know about glycolysis, the whole picture. Let's move on to the next slide, and I'll show you specifically how substrate level phosphorylation works to make ATP. On the last slide, I reviewed the energy payoff phase of glycolysis, the last 5 chemical reaction steps that result in making pyruvate and making a little bit of ATP, etc. So here is a slide that's showing you how substrate level phosphorylation works. So ATP is synthesized in 2 different ways in cellular respiration. And one of the ways is called substrate level phosphorylation. And that's what we're reviewing here because it happens in glycolysis. The other way that ATP is made is by oxidative phosphorylation, and that's going to occur in a few slides, actually in part 2 of this chapter. So don't worry about that right now. So substrate level phosphorylation occurs in the energy payoff phase of glycolysis. And what happens? So the figure on the bottom is showing you what happens. I described what happens in the last slide, but this is showing you specifically, giving you a picture to visualize and understand. And drawing a picture like this would be fantastic for you to do as a way to get it in your head and understand. So we have an enzyme. and we have a substrate that has a phosphate attached to it. That substrate with a phosphate attached to it fits into the active site of an enzyme. But something else fits into that active site as well, a molecule of ADP, adenosine diphosphate. And that phosphate group transfers from the substrate to the ADP, and out pops ATP. And that substrate that lost the phosphate, well, it pops out of the active site as a product. And that's substrate level phosphorylation. It's a way to make ATP by transferring a phosphate group from a substrate to an ADP in an active site of an enzyme to make ATP. That's the crux of it. That's what happens. The same process of making ATP occurs in the citric acid cycle, which we're going to look at in just 2 slides. Same process. So making it through the last slide means that you made it through glycolysis. You made it through stage 1 of cellular respiration. And I presented to you the details that you need to know and understand. Now we're beginning stage 2 of cellular respiration, which occurs in the mitochondria. And it's called pyruvate oxidation. And there's basically 3 steps that happen. And they're shown here, they're outlined here. And you need to know these 3 steps and what we have at the end of these 3 steps. So this only happens if oxygen is present. Can't happen without oxygen. Well, you're breathing oxygen, I'm breathing oxygen, so this is happening in the mitochondria of your cells, no problem, because oxygen is present, so it can happen. So pyruvate enters the mitochondria. It's going from a higher concentration to a lower concentration through a transport protein. It needs a transport protein to help it get into the mitochondria because pyruvate is positively charged. So it can't get through the membrane all by itself. It needs a helper transport protein to get it in. So that's a concept back from chapter 4 on cells when we learned about membrane transport, that charged molecules cannot make it through the phospholipid bilayer of membranes. So transport proteins are needed to help them get through. Here's an example of it right here. So when that pyruvate moves into the mitochondria, step 1 is a carbon and 2 oxygens is taken out, it's taken off, and that's carbon dioxide. So here's the first time that we're seeing a molecule of carbon dioxide being removed. That's going to diffuse out of the mitochondria into your cytosol of your cells. It's going to diffuse into your bloodstream. That carbon dioxide is going to be brought to your lungs and you blow it out. So all the carbon dioxide that you're exhaling when you blow air out of your lungs is coming from inside your mitochondria. And this is one of the steps where it's being produced. Step 2 in pyruvate oxidation is the remaining little 2-carbon molecule is oxidized. Electrons and hydrogen are removed and NAD is reduced. So we're making a little more NADH to shuttle electrons to where they're going to be used to make a lot of ATP. And the final step is a coenzyme called coenzyme A latches on to that little 2-carbon molecule that's left over. That molecule is called acetyl. And that coenzyme helps the acetyl to enter the citric acid cycle, which is also going on inside the mitochondria in the matrix. So those are the 3 steps that occur after pyruvate moves into the mitochondria through this transport protein. A carbon dioxide is released, electrons are removed and given to NAD to make NADH, and coenzyme A attaches to this little acetyl molecule, making it what's called acetyl-CoA, coenzyme A. There's our 3 steps of pyruvate oxidation. And the last thing we're going to cover here in this video in part 1 is the citric acid cycle. And so here it comes. So a few slides back, actually it was slide number 6, and this is slide number 11, showed you an overview of cellular respiration. And stage 1 was glycolysis. And then stage 2 was inside the mitochondria, and it included pyruvate oxidation and the citric acid cycle. The last slide, we just looked at pyruvate oxidation, so now we're going to finish stage 2 by looking at the citric acid cycle. It's an 8-step chemical reaction. So there's 8 chemical reactions. There's an enzyme that helps along each step of the way, but I'm not showing you a diagram or a figure that shows each of those 8 steps. It's not necessary. I'm showing you a diagram or figure that's basically giving you the summary of what you need to know without diving into the nitty-gritty step-by-step chemistry. And the overall upshot of the citric acid cycle, just to tell you up front, is to finish removing the electrons from acetyl, from the last little molecule that has been produced after beginning with glucose in the very beginning. Now we have 2 acetyl molecules that are going to be processed. And the whole point of the citric acid cycle is to remove the electrons from acetyl and give them to these carrier molecules to shuttle them to the final phases of cellular respiration, where those electrons are going to be used as an energy source to make a whole bunch of ATP. Removing electrons from acetyl is the primary point of the citric acid cycle. So it begins with a molecule called oxaloacetate, which is not shown in this diagram, but the acetyl binds to that oxaloacetate and it becomes citrate. So that would be like right over here. And I'm sure your textbook has a more detailed diagram that would show this. I'm just not showing it on this slide. And that's why it's called the citric acid cycle, because citrate or citric acid then gets processed to remove electrons. And at the end, we get back to having this oxaloacetate that then is like recycled. So 3 NADHs form through 1 citric acid cycle and 1 FADH2 through oxidation and reduction reactions. So you can see that NADH is made, FADH2 is made, NADH is made. So, whoops, this said on the slide 3, but 2. Fix that. I just noticed that. Now, why is the number 2 here? So you see 2 carbon dioxides, because when those electrons and hydrogens are removed, again, carbon dioxide is the waste. So this is where the rest of the carbon dioxide is produced in cellular respiration. We just saw some produced at pyruvate oxidation. Now we're seeing the remaining carbon dioxide is being released during the citric acid cycle. That's our waste molecule. So that's being made. NADHs and FADH2s are shuttling the electrons away. And finally, a little bit of the energy that's being released through this whole process is used to make ATP by that process called substrate level phosphorylation. Okay, what are the net results of the citric acid cycle? Well, the net results are that, well, the answer, there are 3, fix this again, NADHs. There's 2 over here and there's 1 over here. So 3 NADHs are made from 1 citric acid cycle. 1 FADH2 is made, okay? 1 ATP is made. And lastly, 2 carbon dioxides are released as waste for each acetyl. But remember, how many acetyls are coming through and going through this process from our original glucose? 2 of them. 2 of them. So since 2 acetyl molecules go through the citric acid cycle, CAC, for every glucose, then you got to double the numbers. So in reality, starting with 1 glucose, there's 2 citric acid cycles, and in total, 6 NADHs would be made. Just double these numbers. 2 FADH2s, 2 ATPs, and 2 carbon dioxide. By this point, our original glucose molecule has completely been broken apart. The electrons have all been taken out and given to these electron carrier molecules. And a little bit of ATP has been made on the way, but not very much. Most of the ATP is going to be made during oxidative phosphorylation, which I'm going to show next in part 2.